Hello everyone, thanks for watching this video. My name is Shahira Abu Samra. I'm a PhD student at Stony Brook University and I'll be presenting our paper, Topology-Guided Multi-Class Cell Context Generation for Digital Pathology. Pathology data annotations are limited, mostly because they can only be collected through experts. In this work, we propose a method to generate synthetic data that can be used for data augmentation. In pathology, the distribution of different types of cells is discriminative, and so we design our method to create synthetic cell layouts that not only look realistic, but are also conditioned on real spatial and topological patterns. We achieve this by training a generative model to create multi-class point sets of cells, such that their spatial distribution satisfies the spatial and topological characteristics of reference real data. With our method, we generate realistic labeled cell layouts, which can be transformed into pathology images conditioned on reference textures. The generated data can augment training for improving performance on downstream tasks. Limited annotations is a known issue in pathology, whether it was dense annotations, which requires pathology experts to do expensive and time-consuming labeling, or slide-level labels like cancer survival datasets. One solution is to augment the training data with generated labeled data. This usually involves two steps: generating the spatial layout of cells and filling in the stains and textures. Existing methods mostly focus on the second step, but here we focus on the first one, generating realistic cell layouts from existing data. And that's because the spatial distribution of cells, or the cell context, is important for pathology data analysis. It is discriminative in downstream tasks like cell classification, cancer diagnosis, and survival prediction. It is essentially the arrangement of cells in a tissue sample and how different types of cells coexist and are spatially distributed. It is in large part what pathologists examine when they inspect a histology image. A major challenge is the complexity of the cell layout and how to model and learn their underlying distribution. And so we borrow from the mathematics literature to describe configurations of cells. To do that, we needed to answer this question: what makes an effective cell configuration descriptor? We want to capture structural patterns such as clusters and holes, and these are topological features. We also want to capture the spatial colocalization of different types of cells, and these can be captured by spatial statistics. Clusters and holes are topological features. We use persistence homology to capture and characterize the topology. With persistence homology, we apply filtration on the point set of cells. The filtration simulates the growth of disks centered at each cell. Throughout the filtration process, different holes will appear and eventually will seal up or die. The distance between the birth and death of a hole marks its persistence and is correlated with its size. And so the persistence homology captures all these holes and encodes them in a persistence diagram, where each point represents one hole and its coordinates are the birth and death times. Now, to capture the spatial colocalization, we use the cross-K function or replace K. It describes the distribution of one class of points in the neighborhood of a source. It computes the expected number of neighbors at different distances from the source, and it can characterize whether the distribution is clustered or dispersed. We use it to describe the colocalization of different classes of cells and also to characterize the neighborhoods surrounding holes identified by the persistent homology algorithm. Now, in order to enforce that the generated and reference cell layouts have similar spatial patterns, we need to first find a match between the generated and reference layouts. To do that, we try to match the holes by their size and spatial context. In this example, we match the holes that have similar surrounding cell context. And so we use the persistence and cross-K functions to characterize and match the holes. Now that we have established a matching between the generated and reference layouts, We want the matched locations to have neighborhoods with similar spatial context. And to train the model to do this, we need the differentiable loss. We compute density maps with different scales and across different cell types, and apply the cell configuration loss. The cell configuration loss will minimize the difference between the density values of the generated and reference layouts at the matched locations. Here are some examples of generated cell layouts. If we look at the generations, last row, and compare with the reference data, we see that they share similar spatial and topological patterns. We evaluate the generated cell layouts by comparing to the reference layout cross-key functions and persistence diagrams for different cell types. Our proposed method achieves lowest error. We show that the generated cell layouts can be transformed into pathology images conditioned on texture data. Here we have a sample cell layout and different reference textures. The generated images have similar texture as the references and similar cell layout as the sample. Additionally, the cell's morphology and textures agree with their corresponding cell types in the cell layout. We use our method to generate labeled augmentation data for use in the cell detection and classification task. Using our generated data improves the model performance by up to 12%. In conclusion, for the first time, we explicitly model and learn to generate realistic multi-class cell layouts with desirable spatial and topological configurations. We propose a novel cell configuration loss where we use spatial statistics and persistent homology to model the spatial distribution of cells. Our method creates realistic spatial and structural patterns in the generated cell layouts, and we believe that the added realism is what leads to improved performance on the cell classification task. In the future, we plan to explore modeling more complex structures like glands, and using our method for improving performance on other downstream tasks like survival prediction. Thank you so much for listening, and please come to our poster.